Hello everyone. In this video we are going to start with our topic number 5, that is internal organization of primary plant body. And in this topic we are going to discuss regarding internal structures of dicot plant as well as monocot plant, in which we are going to study the dicotyledon root, dicotyledon stem and dicotyledon leaves internal structure, as well as we are going to discuss regarding internal structure of monocotyledon root, stem and leaf. Now we have already studied regarding different types of tissues inside the plant body. So these different types of plant togetherly form different structures and organs of plant body. And let's see how these tissues or cells are arranged in different parts of plant. So see, plant body develops from embryo into root and stem. When we sow or when we imbibe the seeds, we see that there is a sprout when seed germinate, and which gives plumule and radicle. The plumule develop into a stem while radicle develop into the root. So this root and stem forms a continuous structure of plant body that is called as a axis of plant body. Here the vascular structure is in continuous form from root to the stem. Now the epidermis, cortex, endodermis, pericycle etc. are continuous in the two organs, but the arrangement of vascular bundle is quite different in the two organs. The stem possesses collateral vascular bundles with endarch xylem, whereas the root possesses radial vascular bundle with exarch xylem. So let's see in detail what is the internal structure of dicot and monocot plants, starting with internal structure of dicotyledon root. See, a cross section through a dicot root in a primary state of growth shows the following tissues from outside to the inside. So first tissue, that is epiblema, which is the outermost single layer compactly arranged protective layer of root with few root hairs. Second one, that is cortex, which is parenchymatous in nature and main function is to store food material and translocation of water and solutes. Third, that is endodermis. Endodermis is the innermost layer of cortex, compactly arranged, having Casparian strips on the radial walls. Fourth, pericycle. Fifth, vascular tissue. And sixth, pith. So all these internal tissues are shown in this diagrammatic representation, and let's discuss regarding each of the tissue in detail, starting with the first, that is epiblema. This is the epiblema. So look at the epiblema. This is the outermost layer of a young root known as epiblema or rhizodermis, or it is also known as piliferous layer. It is single layered. The epiblema cells elongations are tubular and unicellular extension called as root hairs. So these are the root hairs which are the elongation of epiblema cells. Here the epiblema cells has a little bit elongated and look like a hair structure. That's why it is called as a root hair. Root hairs absorb water and minerals from the soil. Secondly, second part, that is cortex. This one. So this region, whole region is called as a cortex. It is multi-layered and composed of parenchymatous cells. The cells are arranged regularly, both radially and in concentric circles. The cells of the cortex are thin-walled with intercellular spaces. Here we can see there is a space between cells. So the cortical cell often contain starch. As we know that cortex has a main function to the main function is to store food material. In dicotyledon root, the cortex stores starch grains. Chloroplasts are absent in the cortical cells of root. The innermost layer of cortex is known as endodermis. Look at here, this layer, the innermost layer of cortex, this one is the endodermis. Now, the main function of cortex is storage of food material as well as translocation of water and solute to the conducting elements. That is transfer of water from cortex to the strands, xylem strand. This is the function of cortex. Now, next part, that is endodermis. Look at the slide. This one, this region, this circular dark color region shows the endodermis. This endodermis is in single layered and well differentiated from the innermost layer of cortex. The cells are living and are characterized by presence of Casparian strips in their walls. So, cytoplasm of endodermis cells is always in contact with Casparian strips, and the Casparian strips are band-like thickenings of suberin material, and the deposition of suberin is restricted to the middle lamella of radial walls. So, the thin-walled passage cells are also present opposite to the protoxylem. In endodermis there are two types of cells with the Casparian strips as well as thin-walled passage cell, and these passage cells are always present opposite to the protoxylem, and which are responsible for translocation of water from cortical region to the xylem strand. The close contact between cytoplasm and Casparian strips controls the movement of material in the root. Water and mineral can pass through passage cells which ultimately reach the xylem strand. Now, the next region, that is stele. The stele is differentiated into pericycle, vascular bundle and pith. Now, this is the pericycle. Below the epidermis or inside the epidermis there is a single layered pericycle which is having thin walled and parenchymatous cells. It lies just below the endodermis. The pericycle is capable of meristematic growth and it gives rise to lateral root primordia and parts of the vascular cambium. Now, next part, that is vascular tissue. Look at here, the phloem, protoxylem, metaxylem togetherly form vascular tissue. This is the vascular tissue inside the pericycle. The arrangement of vascular tissue is in Dicot is alternate and radial. The phloem is not present external to the xylem, but it alternates radially with the xylem. That means the xylem present on one radius, while alternately phloem is also present on the another radius. So the xylem and phloem shows the alternate and radial arrangement. The primary xylem and primary phloem appear as separate bundles and are present on different radii. Xylem strand in dicot root vary in number, that is 2 to 8, and are designated as diarch, triarch and maybe tetrarch. The xylem is exarch, that is metaxylem facing towards the center and protoxylem located near the periphery. Metaxylems are wider and protoxylems are smaller in size. Protoxylem lies next to pericycle and opposite to passage cells. Now look at here. Here the small cells of xylem are called as protoxylem and these large cells are called as a metaxylem. These protoxylems, this protoxylem are arranged in such a way that they are lies next to pericycle and opposite to passage cell, where there is an entry of water and minerals from the passage cell to the protoxylem happens. So now the phloem patches are present alternately with the xylem strand. These are the phloem patches. Look at here. These are the phloem patches. This one. So here the xylem, here present phloem. So it shows the alternate and here xylem and phloem. It shows alternate and radial arrangement of vascular bundle. Now the phloem strand consists of sieve tubes, companion cell and phloem parenchyma. And the parenchymatous conjunctive tissue occurs in between xylem and phloem. Here, this is the conjunctive tissue which is present in between xylem and phloem. Now, at the center in dicot root, the parenchymatous pith is present, and this pith is very small. In old root, pith get obliterated due to secondary growth. So this is all about the internal structure of dicotyledon root.